Protein synthesis. Protein synthesis is governed by this central dogma of biology. Here, DNA are the instructions, and here, protein is the effect. Okay, the effect of that instruction. All right. Now, DNA is found in the nucleus of the cell, and proteins are made on the ribosomes, which are in the cytoplasm. Now, DNA is in the nucleus, inside the nuclear envelope, and can't move. The ribosomes are outside in the cytoplasm, so we need a messenger to carry the instruction from the nucleus to the ribosome, and that is mRNA. That is the role that mRNA takes. So this process has two main phases: the DNA to mRNA, which is called transcription, and the mRNA to protein, which is called translation. So transcription takes place in the nucleus, whereas translation takes place in the cytoplasm. So let's start off by talking about transcription. It's very important to remember that the only part of the only part of the DNA, the genome, that will be transcribed is the gene in question. Okay? So of the 20,000 odd genes in the nucleus, only a single gene that is currently being talked about will be transcribed. So what has to happen is that the DNA unwinds and unzips in the region of the gene. So that's happened here, and an enzyme does that. That enzyme is called DNA helicase. So that enzyme unwinds and unzips the gene. Then another enzyme called RNA polymerase comes and lands on the template strand of the DNA. It doesn't just land at random, it binds to a specific region at the start of the coding sequence. And that enzyme is called RNA polymerase. Now, as its name suggests, the role of RNA polymerase is to make RNA. And so as it moves along the template strand, it brings in the complementary RNA nucleotides to this DNA code and gradually builds up a RNA molecule. Now, as you can see, as it's doing this, it's using the same base pairing rules that we see in DNA, with one exception. Where we have adenine, in DNA you would bind with thymine, you would pair with thymine, but in RNA you pair with uracil. Okay? So there we have our mRNA molecule, and when the RNA polymerase gets to the end, it falls off, and then the mRNA transcript leaves the DNA. So the temporary bonds are broken. The DNA now is able to coil itself back up again, and the RNA transcript is able to leave the nucleus. So that is transcription. So our next stage is to look at translation. So what has happened is, here is the nuclear membrane. mRNA transcript has left by the pores and is now in the cytoplasm. So the first thing it needs to do is it needs to associate itself with a ribosome. And ribosomes, as you will remember, have two subunits, and the mRNA sits between clefts, and if it's into these two clefts that the next piece of RNA will form, the transfer RNA. Now, transfer RNA is very important because it is this, the tRNA, that brings the amino acids into the equation. So, the first thing that has to happen is amino acid activation, where the tRNA molecule binds to its specific amino acid. What determines which amino acid binds to it is the anticodon loop, so the 3 bases on the anticodon loop, and that is an energy requiring process. So, we now have the ribosome sat on the mRNA, and each cleft is exposed to a codon, 3 RNA codes. The tRNA with a complementary anticodon will come and sit in the cleft. So there's the first tRNA, and there's the second tRNA. What that does is it brings the 2 amino acids close enough together for a strong peptide bond to be formed between the parent of them. Once that bond is formed, the ribosome moves 1 codon along. This tRNA drops off and goes off to find a new amino acid, and the next tRNA in the sequence can come in. A peptide bond will be formed between amino acids, and the ribosome moves on, the tRNA drops off, and so on, until you get to the stop codon, where the tRNA will have no amino acid. So then when that moves on, this drops off, and now your polypeptide chain can then coil itself up and become a protein to be probably sent to the Golgi apparatus, where it will then be packaged up and sent off to the rest of the cell.